These tubes that contain this anticoagulant have a precise amount in them. You can see this light blue in here. It has a little bit of liquid in the bottom. All right, and that is sodium citrate. So you have to make sure you get enough blood in here to spread this anticoagulant out. If there's not enough blood to anticoagulant ratio, it'll mess your results up. So these have to be filled up all the way. And technically, if you can put 10 parts of blood in here, this would be 1. So it's 9-to-1 in this tube, which is a test question, hint, hint, hint. Okay. You gotta make sure you fill them up completely and mix them well. Can be over diluted or anything like that. What could happen is you could get tiny microclots in these tubes, and then that could gunk up your machines. One thing. All right. Which one of these do we use? How do we know what to use? Well, it depends on what test we want to look at. Okay. EDTA for whole blood is good because it doesn't affect red blood cell shape too much. It's good for that. It preserves blood cell integrity well. It also prevents platelets from clumping. Do you remember in the clotting or the stages of hemostasis, there was adhesion and aggregation of platelets? It's going to prevent them from sticking together. And it's compatible with blood staining, which is when they'll stain your cells with some kind of pink or purple dye, and you can look at it under a microscope. Otherwise, cells are clear and translucent. You can't make heads or tails of them. So we have to stain them, and this doesn't mess with it. Look on the next page and tell me the first tube you can find that has, on page 129, which one has EDTA in it? Give me hints on page 129. Okay with stains. That C with a hashtag means with. And actually that means without too, so. Okay, so it's purple. My question that I asked about which tube contains it, lavender does. It's the last one in our blood draw, right? So that means that EDTA must really affect other additives or not, or if it comes last in order of draw. It does. So they're going to put it at the end. They don't want it interacting with anything else. But it's still pretty popular, pretty good one. The next one, I'm reading at the bottom right side of 27. This is all like 1 or 2 paragraphs. I'm just really breaking it apart. Okay, EDTA interferes with coagulation studies. So we can't use it before we do a blue tube. Citrate is used for coagulation studies. Okay, and if this tube right here, light blue, is for coagulation studies, that means I'm going to have citrate in it. Okay, blue is for coagulation studies, so it contains citrate. SPS It's special because it's used for blood cultures. It inhibits certain immune system cells. And that's important because when I draw blood to see if I've got some bacteria working in there to make me sick, I want that blood to be in the same state it was at the moment I took it out of my arm versus the moment it's put in the machine to analyze. So I don't want my immune system continuing to kill any bacteria from the moment it's drawn until the time I get it back out to the lab. I need to inhibit that process, and that's what SPS does. Okay, this is for blood cultures. Right, follow me so far? Okay. Haley, you still follow me there?
Yeah.
Okay. SPS comes in this tube right here. The reason I don't have any blood culture tubes out here is because they're a little bit special. They look like, okay, so they're right here. These. This is way different from one of these little tubes. Okay, I can look on here. It says Bacti Alert, Aerobic Pediatric. Didn't tell me the additive exactly that's in it, but it's something like SPS. So if I'm drawing blood into this jar to see if I'm getting bacteria, it's really easy to contaminate the sample with bacteria that might come from my skin, come from an unwashed hand, come from something I touched inadvertently. So I have to make this the first draw. That way I'm not drawing these in this and possibly contaminating it with something that got into there. So when you draw a blood culture, you're actually sterilizing the site with iodine, and you're drawing two of these tubes, which we'll talk about later, why you're drawing two. It's always first, so you prevent any contamination from this other work that you may have to do. All right. Remember, these things we use to draw blood like these butterflies and these vacuum tube holders, these are multi-sample systems. So we're sticking tubes in there. We need to do it once for the blood culture and be done with that. Then we can go forth with other tests. So that's why it's first. Capiche? Okay. Moving on here. It also neutralizes antibiotics. I'll put that up there. That stands for antibiotics. Neutralizes antibiotics. Okay. Heparin is good for blood chemistry. Things like sodium, potassium, different types of gases in our blood, CO2, oxygen. Okay, and heparin goes right here in green. The last one right now is potassium oxalate. It's cool because combined with something else called sodium fluoride, it is used for glucose determination. Okay. Let's go over here and look at page 129 under the gray tube. The gray tube picture is at the bottom of page 129, and the paragraph explaining it is at the top. Okay, for lactic acid measurements, glucose tolerance tests, which is what Haley just had, fasting blood sugars and blood alcohol levels. So if you have a wreck and you're in the ER and I think you've been drinking, I'm going to use this tube to test your blood. Okay, sodium fluoride is an anti-glycotic. What is this right here? Anybody? It's sugar that we need for fuel for our cells. Okay, the process of breaking that down and using it is called glycolysis. Looky there, we're breaking apart glucose molecules. And if we want to inhibit this, because I want to know how much actual glucose you have in your blood, remember, stuff is still going to be working from the time we put it in the tube to the time we get in the lab. We have to stop it at that point. Then we're going to use something that inhibits that, an anti-glycolytic. It's just going to inhibit glycolysis. Okay, don't forget, you may add or not add this sodium fluoride right here. I'm not going to spell it out. Okay. And so if we're going to use a serum sample, you're only going to have sodium fluoride. If you have a plasma sample, it's going to be either potassium oxalate or EDTA, in addition to NaFl. So you can use plasma or serum. That just gives us flexibility. If I draw one, say, oh, I forgot I need to add this glucose test to my panel, you can do that pretty much in any scenario here, okay, if it's not been too long. So gray, we've got potassium oxalate with or without sodium fluoride. Okay, so these are all anticoagulants. Notice we've got a gap for two colors here still. What are those two colors used for? Which sample? Red and gold. Serum. Looking right here under red, it says chemistry, serology, blood bank, specimen serum. I can look under gold Hemogard or the tiger top, red gray, serum. And so if I want to take the clotting factors out of plasma to equal serum, that means I can't have something that's going to inhibit that. All right. And as a matter of fact, I want a clot to form to get serum. So what kind of additives are going to be in these two, Marissa? Or clot activators? Haley, do you need more time writing here? Are you taking notes?
I'm taking note and I'm reading it.
Okay, I can take a snapshot of this if you need it later. Fill in your notes.
Yeah.
I'm going to.
That'll work too. Make sure I got everything written down right.
Okay, I'll slow down a little bit.
All right, thank you.
So now you should be able to at least line up a few tests, or you're getting to the point where you can start lining up a few tests with each color here. And I'm going to turn the page, look at 128. Just to recap about glycolysis, there's an important paragraph here that explains it. Sodium fluoride inhibits glycolysis and can be used for glucose determination. Glycolysis is a cellular reaction used to harvest energy from glucose. So potassium oxalate and sodium fluoride is used for glucose determination. All tubes containing anticoagulants coagulants should be gently inverted 5 to 8 times directly upon collection. Right. Clot activators, that's our other additive. These things promote coagulation. They also have to be inverted. They facilitate a fast clot. Okay, we don't have time to wait 60 minutes, which is the natural time it takes for our blood to clot in a tube on a bench top. With these additives, it takes only about 30 minutes. It turns that time in half. Thrombin is one of them. Okay, so I'll just make a little space here. And if you go back and look again on page 112, I see thrombin down here in the common pathway. After prothrombin, it also activates fibrinogen. Thrombin is a natural clot activator that we've taken and synthesized and put in the tubes to also promote clotting. Okay, but all kinds of things can be used as clot activators. Inert substances such as glass or silica that can promote clotting by providing more surface area for platelets to activate. Remember, platelets, once they start swirling around something or getting close together, they start clumping and activating. So if I put them in a glass tube, that if I zoomed in here with a microscope and looked at the surface of the inside of this tube, it would be really rough and jagged for glass. And that will cause platelets to start clotting as they are just sloshed around in there. Okay. We can also put stuff in this tube like sand or clay that will also cause those things to bump around and start to clot up. Just remember, you gotta invert these two. Any questions about that? Clot activators are pretty easy. Okay. Thrombin's a major one. We don't like to use glass tubes anymore because why? They can break, and if they break, they can also cut us. And if they have blood in them, that's bad news for us, right? We don't want to be changing blood out people. I'm not going to write the gel up here. It's a little bit too much, I think. But it is an additive in tubes in the sense that it's been put into some of these tubes. It's an inert substance whose density is between that of cells and that of blood serum or plasma. All right. When this blood is spun in that centrifuge, or when this tube is, this gel becomes liquid basically. And when we stop spinning, it will solidify back. And so red blood cells are heavier than this gel when it's liquid, and they will sink through it to the bottom. But serum is lighter, and it will stay on top. Okay, and keeping all of our chemistry stuff with it. And so when it stops spinning, it will gel back up, and we have a nice separation of two layers, serum and a clot or whole parts at the bottom. We can draw that out and use it to test really good some chemistry stuff without messing up our machines. In light green top tubes used for determining potassium, the gel prevents the plasma from being contaminated by potassium released from red blood cells. I would highlight that sentence at the bottom of polymer gel. One side effect of red blood cells being broken apart is the release of their contents, and specifically potassium. And so you can infer that if there's extra potassium in my blood sample, I might have false elevated potassium levels, right? So we don't want to hemolyze our cells. Thankfully, whoever invented this vacuum tube system and this multi-sample needle container, color-coded it, made it pretty much easy, okay? Almost foolproof. You just have to come to school here to be able to learn how to use it. Any questions about that? No, nope, nope. Okay. So at this point, Haley, the beginning of the chapter is a lot about stuff that